model system of P aphids, they harbor the gamma proteobacteria, Buchanera aphidicola. As you can see here, depicted in green, are the bacterial cells nested within a single host cell known as the bacteriocyte. The blue within indicates the host nuclei. So given this really intimate relationship, and this obligate relationship between the two organisms, it brings the question, how do they co-evolve and coexist without either lineage being terminated? And one way is a strict vertical transmission from mother to offspring, and in the case of the P aphid, aphid embryos are inoculated during development within the mother, and then they're born alive. And what we know is that these aphids are actually clonal, so that facilitates the direct co-evolving of both nuclear host genomes as well as bacterial symbiont genomes. More recent work has looked into the actual genomic mechanisms that drive this relationship. And in one study, they were able to reveal that actually the host plays a major role in facilitating the completion of different amino acid synthesis pathways. And so now we realize that not only do they work together, they actually are coexisting and cannot survive without one another in a genomic framework. So that begs the question, well, is there interspecific variation in hosts that can affect the regulation of these symbionts? As I said, in P aphids, they primarily go through a parthenogenic lifestyle, so strictly clonal, where mother will transfer their symbionts to their offspring along with their entire nuclear genome. However, many of you may not know, or actually some of you quite are familiar with, that they actually undergo asexual life phase, where in the fall, during shorter photoperiods, this indicates to different lineages that they must undergo sexual reproduction in order to survive to the following year. And so they produce sexual mothers as well as males that will reproduce, and the eggs within mothers are pre-inoculated with maternal Buchanera and then deposited, and then in the springtime, these little eggs will hatch and form the new generation of aphids. Keep in mind though, these new sexually produced offspring are actually a mixture of two different host genotypes that must now co-evolve with a distinct, different maternally inherited symbiont genotype. So, again, in more detail, how do novel combinations of host loci interacting with identical symbiont genotypes impact the host's ability to regulate symbiosis? So in order to answer this question, we performed reciprocal sexual crosses between 4 divergent matrolines. Specifically, each pair depicted here on this Buchanera whole genome phylogeny represents 2 divergent clusters of P aphids found here in North America. Up above, you see one box in red and another box in yellow that have nearly identical symbiont genotypes, but divergent host genomes. And then below, in the totally divergent cluster, we see outlined in green and blue, how it share a very similar symbiont genotype, but again, possibly very divergent host genotypes. And so, after performing these crosses, we hope to measure Buchanera titer levels, so how much of the symbiont does each progeny clone obtain, and then does this correlate with aphid host fitness? I agree. Okay, so for those of you who don't know, I'll go through this rather quickly just to give you an overview. For sexual induction of aphids, here I have my 4 different lines. We see that they experience a reduced photoperiod, and after several weeks, they should be able to produce both sexual females and males. Interestingly enough, after several attempts under multiple conditions, we realized that 2 of our clones are unable to produce males in standard induction conditions. So, we proceeded with the 2 matrolines, at least from 1 divergent clade, and then crossed them with all the other So we performed these sexual crosses in any permutation we could, and then we harvested the fertilized eggs by hand, and then after a 2-month of diapause at 4C to So just as an overview of our experimental setup, here, for example, we have a maternal Austin cured mother in the middle, depicted in green, that could be crossed with either paternal line to perform crosses that result in two distinct ships. Here I've indicated them as AL. So Austin mothers crossed with LSR1, and over here AY, Austin mothers crossed with 5AY fathers. And they produce a cohort of progeny. And these provide us a comparison of within cross comparisons of full siblings that just share both parents and Buchnera, and then a comparison across half siblings where we can look at how other host loci, other divergent host loci, may impact based on the same symbiont background. So the first step was to measure Buchnera titer. In order to do this, instead of obtaining a single estimate for each clone, we divided each major produced progeny line into 3 subclones, and each 3 subclone was measured in triplicate using qPCR to estimate Buchnera titer. Buchnera titer is the ratio of Buchnera genomes relative to the number of aphid genomes, and in this study specifically, we used GROW-ES to represent Buchnera genomes and EF1 alpha to estimate host genomes. And so again, we performed this using 3 biological replicates as well as 3 technical replicates within to get an accurate estimate of Buchnera titer. And what we see here, let's just focus on the axes first. Along the x-axis are a combination of all the different crosses performed, so the different SIP-ships, as well as their originating parental lines abbreviated. And along the y-axis is you see a wide range in titer value for some more than others, which is quite interesting. So at a first go, just looking at titer and how that responds after sexual cross, we see that titer levels significantly differ between offspring and parental clones, as well as between half-sibs, demonstrated in the first 3 columns. Here you have an Austin mother, right, who shares the same symbiont genotype and half of the genome as her offspring in AY as in AL. However, we see a significant difference. So that implies that the host genotype is actually playing possibly a significant role in determining and regulating symbiosis in this system. However, does this variation have any consequence? Maybe it's just noise, who knows? So we wanted to go in and test this by looking at aphid host fitness. So using the same design, we, for each of the 3 subclones representing each progeny line, measured time to adult, which is days, how long do you take to develop, how big are you, time to reproduction in days, so when do you actually start reproducing? Keep in mind, these are not clonal lineages, so the sooner you reproduce, the sooner you can get more of your own clonal offspring out there. Also, number of offspring produced in 6 days, and that's because in the P aphids, the first 6 days tend to, that is when most of the offspring are laid. And then lastly, we also estimated reproductive rate, which is a proxy to look at how many aphid progeny are produced per day per clone, per given clone. And what we see is that aphid fitness actually does vary significantly across clones, or across SIP-ships as well as clones. And for every measurement I measured, for every clone, in every lineage, we saw significant differences in fitness values across all progeny clones. But now that we see this variation, it begs the question, does it even relate to Buchnera titer? Does the symbiont level have anything to do with this? So we performed some correlations to look at the link between Buchnera titer and aphid host fitness. And so when you have a higher symbiont titer, it seems to be correlated with, when you look at days to first reproduction, we see a positive correlation. So the more Buchnera you have, for some reason, it takes you a little longer to start to reproduce. So that implies some level of slowed growth as well as development. Additionally, if we look at the number of progeny, we see that as you increase your Buchnera load, you actually have a reduction in the number of progeny produced over that critical 6-day period. So lower number of offspring. And last but not least, as a nice a reduced reproductive rate. And from a clonal species, that is very important. So, in summary, for our question, is there interspecific variation in hosts, and does this impact the regulation of symbiosis? We can answer that symbiont titer levels differ between offspring and parental clones, and that this titer level also differs between half sibs, so those that are actually relatively genetically related. We also find that a higher titer level is associated with slower growth and development, as well as a lower number of offspring. So, in synthesis, with all of these facts, we find that following sexual reproduction, sibling aphid lines actually show a significant variation in titer levels and overall fitness, and that this might potentially actually reflect a cost to regulation